Most of us have probably used a biometric facial recognition system. You know, the one where you take a selfie like the most obnoxious person there is and it unlocks your device. What if we had a similar system but for cancer cells? What if we could recognize specific cancer cells by just taking a picture? Most importantly, why do we want to build such a system? In today's 3-minute paper, we'd like to discuss a study which aims to develop a deep neural network to do exactly this. A 2009 editorial in Nature pointed to disturbing results of cell testing which show that 18 to 36% of cultures used in scientific research were misidentified. It is an identity crisis of a different kind. There are many reasons why cells are misidentified. For instance, they may have been swapped or contaminated with other cell lines. The consequence can be catastrophic. Imagine a cancer researcher tests a new drug on cells which are not what they think they are. We can use short tandem repeat (STR) analysis, which is the gold standard method based on genetic testing to identify cancer cell lines. But there is a but. Suppose I have a cancer cell called COLO704. I treat the cell with an anti-cancer drug, and the cell develops a resistance. Since drug-adapted COLO704 and the parental cell have the same genetic makeup, STR cannot tell them apart. It's like trying to tell identical twins apart. We adapted a convolutional neural network, CNN, to solve this problem. We use a CNN which has been trained already on ImageNet, a dataset of 14 million pictures of everyday things. We then retrain this CNN using our much smaller cancer cell dataset. It's a bit like retraining a worker who already has 10 years of experience in coding to learn a new computer language in just 2 months. Our best model achieved an average F1 score, a measure of a test accuracy, of 94% for the 4 parental cells. For both parental and drug-treated cells, our best model achieved an average F1 score of 91%. Furthermore, our model is very confident in predicting the correct cells with an average probability confidence of 92% for true positive compared to just 64% for false positive. From these results, it would appear that drug-resistant formation is associated with changes in the cell structures, making drug-adapted cell lines distinguishable from the parental cell lines by our CNN model. Note that the gold standard DNA-based method STR and most human experts cannot make this distinction. This approach can provide a low-cost method that enables the monitoring of cell line identity as part of the daily laboratory routine and also the authentication of genetically identical cell lines. Thanks for listening.